And I spoke at various places all across this campus and the med school place. So I actually wanted to talk about something new, but I didn't have the opportunity to put it all together. So this is something I talked about at least once or twice last term. The last time I spoke somewhere around here, I talked about marijuana, its benefits of using it. No, I didn't want to talk about that one, so I put this together. And actually, I think Chris asked me like the day before yesterday. Then I went to Jessica and said, "Hey, Jessica, I really don't want to do this now because I'm really busy and I have other things I would like to get done. And what I would do is come back later. And what I really wanted to do after talking to Dr. Barry and Dr. Gaffney was put together something such that I would say like about a career in the pharmaceutical industry. What does it take? What do people in that industry look for in prospective candidates? What kind of people in that industry get jobs? And what kind of jobs there are?" But Jessica said no. So here I am. So what I want to tell you that a little bit about drug discovery and how organic chemistry is really at the crux of the matter. Almost something like 98% of all drugs that are used today are small molecule inhibitors. Okay, and that means small molecules means usually organic chemistry oriented, right? There's a few inorganic chemistry type stuff like cisplatin for cancer and stuff like that. But by and large, it's all small molecule stuff. And so I'm going to tell you about that in terms of organic chemistry. And this project is called Tissue Factor 7A. The idea is to develop an orally administered small molecule antithrombotic that has minimal bleeding side effects. That's the mechanism, inhibition of that complex, and this is for treatment of chronic coronary artery diseases such as infarction or angina. But there's other indications that it could be useful for, like stroke, embolism, peripheral artery disease, deep vein thrombosis. Collectively, globally, if you think about anything that you're required to buy to treat heart disease, first of all, heart disease is easily the largest killer in the Western world. If you take the 1 through 10 killers, top disease killers, then over half of them, you total those all up, that will only equal half the amount of people that will actually die from some form of heart disease. So the global market for this, if you think about buying things, including over-the-counter, like aspirin, to a prescription, that market's roughly around $15 to $16 billion a year. So it's big business. And it's important to be chronic because share owners want a return on their investment. And the idea for a drug company, the perfect drug would be this, is that the day you're born, you start taking a pill a day, and you take it as long as that pill's covered by a patent. And that drug company will make tons of money doing that. That's chronic. So this is my overly simplified cartoon of an artery. Kind of looks like a piece of PVC pipe. But everything that's required to form a clot is circulating in your arteries except for one thing, and that's tissue factor. Tissue factor is expressed in plaques, which are derived primarily from two different ways. One's diet, in which you control, like eating pizza is a good way of forming plaques on your artery. The other one is from your good old Uncle Ed, and that's a genetic predisposition. In fact, babies who are born have plaques on their walls, and that has some other implications, but it's always there, okay? And you can control it to a certain extent through your diet, but you can't control the genetic part, okay? Now, when a plaque ruptures due to some type of injury, like if I hit you, or maybe the atrial contraction causes reverberation, causes a plaque to rupture. Tissue factor is exposed to the circulating cofactor. In this case, tissue factor, in this case 7A, they combine and initiate coagulation that results in the end in a thrombus. And this is actually what is called atrial thrombosis. Okay, I should also say that I have a funny word, thrombogenicity of plaques correlates to tissue factor levels. That's just a fancy way of saying the bigger the plaque, the more tissue factor is present. Okay? So this is my dumbed down cartoon of what the coagulation cascade looks like. So after this, first of all, I should say that all of these enzymes in here belong to a superclass of enzymes called serine proteases. There's about 4,000 known in the human genome. So digestive ones, for example, like trypsin and chymotrypsin are also serine proteases, and I'll show you what makes a serine protease in a minute. But once you form this complex, this tissue factor 7A complex, it proteolytically activates factor 9 to factor 9A, factor 10 to factor 10A, which then in turn activates prothrombin, which is known as 2 to 2A thrombin, and then thrombin cleaves fibrinogen, which is known as 1. It kind of depends on which literature you read. If you're a chemist, you call them by this name. If you're an enzymologist, you'd use the numbering system to 2A, and then fibrinogen to fibrin, and that's 1 to 1A. And these then, fibrin, then combines with activated platelets, which results in the formation of a thrombus. And these activate, that's what I was trying to describe here. So it looks like the fibrin is these little squiggly lines, this protein mesh. The platelets are these things on the top. So imagine taking like some type of fishing net and putting fish in it. That's kind of what it looks like. And that restricts the flow of plasma and stuff through the system, and that results in an occlusion. And the occlusion, based on where it's at, if it's in an artery leading to the head, that results in a stroke. If it's an artery feeding the lung, that's an embolism. If it's in artery in the leg, that's a thrombotic event. If it's in the heart, that's an infarction. Okay? Now, currently there's, well, I'm going to fib. I'm going to lie to you and say there's only 2 orally available antithrombotics on the market. Really, there's 3, but the third one is only restricted for people, you have to be an adult, you have to undergo knee or hip replacement surgery. If you don't fit within those 2 parameters, then you're not allowed to take it. So for all practical purposes, there's only 2 orally available antithrombotics on the market. And they both have issues associated with them. The first one is called Coumadin, which I have written here as warfarin, and I'll explain why in a second. Now, Coumadin has lots of problems associated with it. It has food effects. In fact, they have a book that you can go, if you go, do they have Borders here? Or Nobles? What kind of bookstore do they have around? Barnes & Noble. Okay, so they have Barnes & Noble. You can go to Barnes & Noble and buy the Coumadin cookbook. And that's a book that tells you how to minimize food-food interactions when you take this drug. The second big problem with that is you also have a lot of drug-drug interactions. If you're on Coumadin, you're not allowed to take certain drugs because it results in it either being inactivated or it being overly activated. And the last one is that it's a bleeding side effect, and that's the reason why I use the name warfarin up here. You can go down to Walmart's big here. You can go over to Walmart and you can buy Decon rat killer. So this is a human drug, but it was first marketed as a rodenticide to kill mice and rats. You put out a little plate of this junk that's laced with warfarin. That's what you call it if you go down to Walmart. They eat it, they run along the wall, they bump into the wall, and then the rat bleeds to death internally because it can't clot by taking that drug. And just keep that in mind because, there's probably somebody that you've known that's actually taking it. Right? You're taking a rodenticide. That's kind of a weird thing to think about, right? So that's probably not the safest thing to take. Because if you take a little too much, you bleed to death. You don't take enough, you have a heart attack. The second one is called Plavix. Everyone's seen those Plavix commercials? And the problem with that is it's an irreversible P2Y12 receptor inhibitor. And what that means, the only way that you can have a rescue therapy is through complete blood transfusion because it takes about 23 to 28 days for your blood to recycle, to regenerate itself. So, for example, if you're on Plavix and you schedule your wisdom teeth to be taken out next Tuesday, you'd actually have to, they'd make you wait for about another month until this Plavix bleeds out of your system. More importantly, if you're in a car accident and you're on Plavix, right, and you have some type of contusion or you're cut or something like that, that can be a life-threatening issue. So back in the early, think about this, I think the late 1990s, people did some pharmacology studies using protein-based inhibitors of various enzymes. The first one was tick anticoagulant protein, which was extracted from those ticks that feed on you and the dogs. They used this to inhibit factor Xa. Then they used hirudin, which was derived from freshwater leeches, to inhibit thrombin. And when they did these pharmacology studies inhibiting these enzymes, there was always excessive bleeding associated with them. So what we did was took an enzyme derived from the Kunitz domain of factor 10, which inhibits factor 7A, and you did that exact same study, there wasn't excessive bleeding. Now, intuitively, you'd think that makes perfect sense because by design, these parasites need to feed in a certain fashion or whatever. And you expect, like, if a tick is going to bite you, it's going to feed, and so you're going to be bleeding the whole time so it can eat, right? And they targeted specific enzymes, evolutionary-wise, such that you're going to target an enzyme that's going to result in bleeding. So they didn't target 7A, and so inhibiting it, you don't see any bleeding. So you kind of expect that. But you had to perform the experiment to be sure, okay? Now, this disparity between all these other guys bleeding and the other one's not, led us to believe that this presents an opportunity that you can prevent amplification of this coagulation cascade prior to coagulation, because if you think about it, the turnover number for tissue factor 7A is about 100,000. So enzymatically, that's kind of in the middle of the road. It's not that great, but it's not that bad. But it will convert 110 to 10A 100,000 times. So it generates 100,000 molecules of that. And then let's say it does the same thing. 10A takes one thrombin and converts it to 100,000. One prothrombin and converts 100,000 to 100,000 of those up for thrombin. Okay, so you start with one, you end up with 100,000, then you have 100,000, then you're up to about what's that? 1 billion, then 1 billion. It gets amplified really quickly. So you don't want to do that in the end. And then the other thing is that the safety window is going to get much wider than what you've seen before, because these other ones always cause bleeding, right? And you want to be able to take the drug. You take a little bit too much, you're not going to bleed to death. You don't take enough, you're still going to get no clotting. And so that therapeutic window is getting widened. That you don't see that by inhibiting those other enzymes. So in I think this is 1996 or 1997, so somewhere in the mid-90s, there was a guy named David Banner. Has anybody ever read comic books? Huh? That's right, the Hulk. So Dr. David Banner, the radiologist, gamma ray, right? He takes an overdose of gamma radiation, and when he gets mad, he turns big and green. But it's not the same guy. This is a different guy. He was at Hoffman-LaRoche, and they published the very first crystal structure of tissue factor 7A with this enzyme, which is called PPAC. And PPAC stands for phenylalanine, another phenylalanine. BPA is arginine. If you were a biochemist, those would be the wrong letters, because arginine is really an R, right? Phe's right. And then chloral, let's see, methyl ketone. That stands for PPAC. Got the co-crystal structure, and this is what the structure looks like. And this is how we're going to define to kind of illustrate what a serine protease is, okay? Because this is always common in all of them, this one motif. So first of all, you have to look at this part. This is really how you define the enzyme class. This is a serine, hence serine protease. And this serine attacks this carbonyl group. And then it's called a catalytic triad because it involves a serine, a histidine, and then you really can't see it in this picture, but I'm going to show you a few more later. Right behind it is an aspartic acid, and it shuttles the proton away from this group right over here. Then you have this thing called an oxyanion CoA, where you have two backbone glycines, the NHs from these backbone glycines, that stabilize the alkoxide, the oxygen anion of the tetrahedral intermediate. Then you have if you're an enzymologist, this is called an S1 pocket. That's the substrate 1 pocket of the enzyme. And inside that, at the bottom of that pocket, there's an aspartic acid, aspartic acid 189, that forms an ionic bond with the arginine in kind of like a head-on-head fashion. So it's a pretty tight binding situation. Then you have this peptidic ridge, and if you notice, you have these NHs, and they kind of bind in a beta sheet fashion to that peptidic ridge. You have a lipophilic S3 pocket and a lipophilic S2 pocket in this as well. Now, if you're speaking in terms from the enzyme, these pockets are called substrate pockets. If you're talking about it from terms of the molecule, it's called P, because for peripheral. It's the thing in the molecule that sticks into the pocket. Okay? So it's P1, P2, P3 in terms of the molecule, S1, S2, S3 in terms of the enzyme. There's actually an S4 substrate. Beyond the catalytic triad, it goes prime. So it's S1 prime, S2 prime, S3 prime. Serine proteases always do bond cleavage between an arginine and some lipophilic amino acid, typically is valine. That's the typical sessile bond. So I should say this. We took this enzyme structure like 5-6 years later. We overlaid it, compared it to known thrombin structures. And there's actually a surprising difference between the two. This little loop here is called a insertion loop. In 7A, it sits over the catalytic triad. In thrombin, it sits over the S2 pocket. And what that does is restrict the accessible space into the S2 pocket. Kind of makes sense because thrombin is actually involved in other biological pathways, predominantly the arachidonic acid cascade. Anybody heard of that? It's involved in inflammation and cancer. So it needs to be more promiscuous, I suppose, yeah. But what that led to the idea that maybe we can determine if that's a mode of getting selectivity versus that one particular serine protease, which is highly homologous with this one. In order to test that idea, we decided to prepare a library of trimers using the same trimer, amino acid trimers that you'd see in PPAC, except we want to make a few changes because it's easier. One is you swap out the chloromethyl ketone with a ketothiazole. Ketothiazoles are easier to make. They're slightly less electrophilic. Alpha chloroketones, they always undergo disproportionation to generate ketene, chloride, and the other piece, whatever it may be in that case. So swap this out to make it easier to make, and you can store it in a jar. Then we're going to change this middle one and vary the size of this group here so you can see how big you can get into the S2 pocket and still be tolerated while you're trying to prohibit it from getting into thrombin and run it in both assays. So one it should be active in, and the other one, if it gets large enough, it won't have very much activity in, and that's what you want to see, okay? Actually, this is a common strategy in this area to do something like this. These are called informer libraries, to inform yourselves about the pocket, the size of the pocket, and so on and so forth. Using ketothiazoles, that's been demonstrated before, so that's known as well. And see what we get. And then the whole idea is to take this information, define the size of the pocket, and then make it a non-peptidic core because you can't orally take a peptide. Does anybody have an idea as to why you can't orally take a peptide?
It'll get digested.
That's right, because your stomach's full of enzymes that are proteases, for lack of a better word. You're going to break down all the proteins, so you don't want to take a peptide. That's why when you go to the doctor and get vaccines, they don't give them to you orally, they give you a shot. keep hitting the button underneath this thing, so I'm going to push that in. So here's the library that we generated, and I'm going to draw your eyes particularly to. So this group here is from a proline, and it's small. This is the group that you'd find here. This is the little R guy. This is sticking in the P2 pocket. Then the IC50s are in micromolar. So this is 45 nanomolar, or 0.045 micromolar. If you look at this versus thrombin, you have equal potency against 10A, because we're not doing anything about addressing selectivity issues with 10A. But thrombin, for all practical purposes, these are the same within the assay, constraints of the assay. You're not going to get any better than that. That's essentially the same number. If you look at this one, you have a 200 nanomolar inhibitor, 290 versus 10A, that's practically the same number. However, it's 100 micromolar versus thrombin. That's big. So you get to a certain size, you do get selectivity because that insertion loop prohibits it from getting into thrombin. We've got a crystal structure of that one, and it looks just like Dr. David Banner, gamma radiation scientist guy. You can see this is our crystal structure. Here's the P1 pocket, there's this aspartic acid 189, nice salt ionic interaction. Serine 195, you form this tetrahedral intermediate, but the difference is this is covalent reversible. The other one is covalent. It gets stuck there because there's nothing to leave. This one is reversible. When the tetrahedral intermediate collapses, the enzyme dissociates. Catalytic triad, here's the S3 pocket, here's the S2 pocket, but there's an extra interaction that we see in this one that you don't see in the other one, and that is this interaction through this water molecule to aspartic acid 60. Now that's kind of important because aspartic acid 60 is mutated in 10A, it's an alanine, so it would just be a methyl group. But in this one, it's an aspartic acid, and that's how we're going to try to get selectivity versus 10A by engaging that amino acid. Does that make sense? So, nice enough, you get a big enough molecule there, you can get about 500-fold selectivity versus 10A. That's kind of unique. So we took that molecule and you morph it into something that's non-peptidic. I chose this heterocyclic core. It's called a pyrazonome. It's not a big intellectual leap because if you ever made peptides before, small or big, one of the common byproducts is backbone NH attacking the carbonyl of another one. So like this NH cyclizing and attacking this one, forming piperazines or ketopiperazines. It's a common byproduct, so it's not that big of an intellectual leap to try to make this, morph this. But then you make it non-peptidic by aromatizing it. Then it's not very readily handled by your body in order for metabolizing purposes. You took this molecule, took our crystal structure, and you overlaid them. The gold one is the crystal structure. That's this little yellow stuff sitting here. And the blue one is the molecule that we designed. And you overlay the two, and you can see there's a lot of complements between the two. A lot of complementary overlap. And you just said, okay, let's go ahead and go forward with this. So we set out to figure out how to make a pyrazonome-based inhibitor, a non-peptidic 7A inhibitor. And here's how we did it. I'm going to go over this at a high cursory level, little detail, for two purposes, and they'll become apparent later. But just trust me, I'll get in more detail. So you take benzoglycine, this, throw in an aldehyde and trimethylcyanide. That's called a Strecker reaction. It generates alpha cyanoamines. You cyclize that intermediate to give something that I call chloropyrazonone. You do nucleophilic aromatic substitution. Technically, this is an ipso substitution to add this amine right here. Hydrogenolysis at 60, I'm going to say at 60 psi, you get benzolysis of the ester as well as dechlorination of this chlorine bond. Couple it with this amine that's protected, which has to undergo hydrogenation as well to clip off this protective group to give the inhibitor. Or if you do this at 20 psi, you only get benzolysis and the chlorine stays intact. So you can do it selectively. You can either leave the chlorine on based on pressure, hydrogen pressure, or you can take it off. What the point of this is you go through 1, 2, 3, 4, 5, 6 steps. Each step, you have to isolate, purify, then resubmit it. So that's about 2 days per step. So 6 steps, that takes 12 days to make one inhibitor. As a consequence of taking so long, because it's a linear sequence, it's very slow and a time-consuming process to develop structure-activity relationships. In an ideal world, what you would want to do is this. You take your chlorpyrazole, you dump in one amine, you stir it around for a little bit, then you dump in this one amine, it only reacts where you want. You want it to only react where you want. Then you dump in your second one, and it reacts at the place that you want. And then you remove the solvent, and you have your inhibitor, and you test it, and you get information on your system, right? That would result in it being super fast, getting structure-activity relationships. By the way, you know, in terms of, if you look at what it means to be on the market today versus tomorrow, if it was Lipitor, today I would make $253 million. If I started tomorrow, that means I lost $253 million today. It's a big deal getting there quicker. So in order to get as close to this idealized synthesis, we developed this polymer-assisted solution phase synthesis. And I'm describing this where it looks like it's just one compound, but typically this was done on 48 compounds at a time, 48 to 96. Okay? Usually you can do them in blocks of units of 12, 12, 24, 48, 96, all the way up to 384. Never went that big because it becomes too cumbersome, but 96 easy because you can make manageable amounts of material. So even though I'm showing it for one, imagine it being 48. And each one of these things equals something different, okay? So you take this chlorpyrazole, you dump in the amine that we're going to be placed at the P3 position of the enzyme. You stir it around in acetonitrile overnight, and in the pot, after you remove the solvent, you would have your desired product, the conjugate acid of the amine, and any excess amine sitting there. Then you redissolve that stuff up in methanol. You dump in potassium hydroxide to do a sponification and ester hydrolysis. And in that pot you'd have the carboxylate salt, the alcohol derived from ester sponification, excess base, the original amine we added in the first step, and a little bit of the salt from this guy. In that same pot, you add in acid, HCl, you acidify everything. That protonates all this stuff, right? Protonates this guy, makes this a conjugate acid of the amine, and now you have this salt. You remove all these solvents and then redissolve in dichloromethaneum methanol. Now, if you notice, this is still done all in the same pot. Nothing's changed yet. There's been no purifications. We've done 2 transformations. So the next step is going to be a purification step, and it's a resin capture release technique, okay? So you take this stuff that's been redissolved with all this whole gamish, you dump in this polyamine resin. It undergoes an acid-base reaction, right? This being a base, this being an acid, and you get this, the desired product is captured ionically on the resin. So you can take this now and wash it with multiple solvents, and all these byproducts are washed away. So all what you have left is just resin with your desired product on it. You take that and you acidify it, and it falls off the resin under acidic conditions to give you pure product. And the average purity on these runs are typically greater than 95% using MSUV to determine that. So then you take that stuff, you redissolve it in another solvent. So you remember, it's showing it for one, but you're doing this on like 48 compounds at a time or 96 at a time. You add in this carbodiimide resin. You add in something called hydroxybenzyltriazole, which is an activating agent. N-methylmorpholine, which is a mild base, and dichloromethane and DMF, stir it around for about an hour, and you generate this activated ester. This is kind of like making a mixed anhydride of an acid. It increases the electrophilicity of this carbonyl right here. You add in this amine. This is the P1 amine that's going to go into the S1 pocket of the enzyme, right? And you generate in this same pot. So in this pot you have your desired product, unreacted acid or activated ester. You have a little bit of this amine that might be left over, and you have HOBT as well as this resin. To that same gamish, you add in this polyamine resin, but this time instead of grabbing your desired product, you're going to remove the undesirables. In this case, the acid or the activated ester, as well as HOBT, it will remove that from the solution. Then you add in this aldehyde resin, and aldehydes and amines react to form amines, so it irreversibly removes this amine, and you just filter it and wash it with a little solvent, and you get your inhibitor, right? Now, oftentimes, these are inhibitors, as you could tell from that one time when I kind of gave you that cursory overview, they're brought in with protecting groups. If it's a carbobenzyloxy, T-butoxy protecting group, if you just incubate it with a little HCl dioxide, it deprotects it. If it has a carbobenzyloxy protecting group on that amine, you have to get a little more fancy. So in the late 90s, early 90s, I should say, Michael Young at UCLA developed these in situ conditions. That's kind of like a Finkelstein SN2 type reaction to take off this group right here. And so what it does is in situ generates TMS iodide. You take TMS chloride and sodium iodide, mix them together, you make TMS iodide. Heat it up and it takes off this protecting group and it generates benzyl iodide. If you use that standard polyamine resin, you can't do that because it will irreversibly recapture this thing through an additional elimination reaction through this thing here. This is like a vanillagous ester or acid. So this amine attacks this carbon here and kicks out ammonia, and you don't want that to happen. So we had to use this one right here, and it actually reacts forming pyridinium ions of that benzyl group. So you're able to remove that, just filter, and you have your pure product. So what normally took about 12 days to make it one at a time, you can make 96 in 5 days. So the throughput is much higher. You get much more information on your system quicker. That's the benefit. So here's some SAR that we generated from this thing. Because, in a couple of weeks, you can make a couple hundred compounds really quick. I'm going to give you the important stuff. First of all, this NH is critical for potency. And that is because, if you remember right, that PPAC molecule binds in a beta sheet fashion to the backbone of the enzyme. If you substitute that, or you set in such a way that you don't have an NH, but like two R groups here, it can't do that. So it won't bind in the pocket anymore. Another one, I should say, if you have to maintain the basicity of that amine. If you don't, it doesn't bind to that peptidic ridge. So adding things like amides or sulfonamides kills the potency as well. And then we found that small aliphatic groups are optimal, and that makes sense because that S3 pockets is lipophilic. So you want greasy things over there. The P1 is much less tolerant of groups. It ends up being, you have to have this benzamidine group here. If you do anything that attenuates the basicity of this, because it's binding in the bottom of that pocket to that aspartic acid 189 in an ionic bond, if you make it less basic where you don't have this ionic interaction, there's no potency whatsoever for the system, okay? So we looked at SAR. There's really a 3-variable problem. You had 3 different pockets that you're wanting to stick things into. We were able to quickly assess around 2 of them, but we were missing one, and that's this S2 pocket right here. And the problem is that from the synthesis that we developed the first time is that you stick this guy in the very first step. The very first step of that 6 or 7-step synthesis, you install this P2, for lack of a better word, diversity group. That's the part you want to diversify so you can get some structure-activity relationships about it. And that's a problem. While it works well for some stuff, it's the functionality, the toleration of this functionality is not all that great. And the reason is, you can understand it in terms of chemistry, and that is unreacted aldehydes fail to undergo aminimide formation, which is required so that you can form this Sprecher intermediate. Then, that's one problem. So you have to have reasonably reactive aldehydes. The second problem is that if you're able to form this Sprecher product, it's oftentimes unstable with cyclization conditions. I didn't show you what those were, but what you do is you reflux it in orthodichloropbenzene with oxalochloride. So you're always generating some HCl. It ends up being the case oxalochloride boils at about 130 degrees. Orthodichloropbenzene, I think, is around 180. So you never actually get to the boiling point of orthodichloropbenzene, but you're actually essentially refluxing oxalochloride. That's kind of harsh conditions. And in practical terms, this is kind of like a vanillus aminal, right? You have a great leaving group right here. This lone pair on this nitrogen, you just push this out, kick out cyanide. That's how disproportionation occurs. So that's bad. All that stuff's bad. As a consequence of these issues, the scope is small. But you want it to be big because you want to diversify all this stuff, right? How am I doing on time? Where's Chris? Oh, there you go. Am I okay?
Oh, yeah.
Because I like talking about myself, and I'm going to keep going until I have to stop.
Yeah, we have room until 1:30, I think.
Okay, good, because that's good. So you think about those problems, you say, hey, when there's a big problem, how do we fix it, right? And the things that you want to have fixed are things like this. You want to install this group at a much later stage in its synthesis, and that makes it diversification friendly, right? You want to be able to quickly access all that stuff later on. In order to do that, you want it to be a mild process because you want to incorporate as much functionality as possible. And then from a business perspective, you want to think about who's this go to. So this is a discovery process. In the end, it's going to go to development. So it's going to have process chemists involved. You want to make the chemistry as process friendly as possible such that they don't have to do as much work because that might save a day getting to the market. And like I said, a day can be hundreds of millions of dollars. So this is called a retrosynthetic analysis. And you work your way back, and this is going to be the stuff that I'm going to start with. And you can see, imagine something like this, where you see a P3 group, P2, and P1, and this is going to be the template that you want. So here's how you start it. So you can take chloroperazone or 2,6-dichloroperazone, this one, heat it up with amine, you do aromatic nucleophilic substitution, right? Ipso substitution. You get this product. If you use the dichloropyrazone, only one amine adds because when you add that first amine, that heterocyclic system becomes too electron rich for a second one to add. Only one can add. So it doesn't matter how long you leave it there, only one will add. Then you do electrophilic aromatic bromination, just stick these two guys on here. Then you do another aromatic nucleophilic substitution, an ipso substitution, but you only substitute at this spot right here. Now, even though this looks like a super simple molecule, to try to ascertain where you got the substitution of the hydroxyl group by spectroscopic means is incredibly difficult because it's so simple. You had to resort to, is anybody taking Dr. Berry's qualitative organic analysis class? Okay. The stuff you're doing where you're making these derivatives in the beginning of these simple compounds, you just left. You know how you're making these simple derivatives of these compounds? Well, doing that stuff's really important because that's exactly what we did here. We resorted to that 1880s technology, made this compound right here, took a mass spec, it had this weight, so you knew it was this compound. If it was one in which the hydroxy added here, it would have been a dimer. It would have been twice the mass. Oh, I'm going the wrong way, aren't I? Okay, so then we took that pyrazine, and we looked at alkylation studies. Okay? We varied the base and solvent. We kept the temperature the same. It was always at reflux. drive this reaction. You get either N alkylation or O alkylation. If it's, here's the ratio. In the end, we found it required a bidentate base. You have a chelation-controlled alkylation process. The calcium binds, it's captured right here, between this nitrogen and this oxygen, blocking the oxygen O alkylation process, only leaving one side accessible. That's why you get this type of selectivity. Now, I have two equivalents there. Really, it's really one, but the problem is with calcium hydride, when you pop the lid, it absorbs water super fast, right? So stoichiometry-wise, if you used a fresh bottle, it was only one equivalent. If you've been using it for a few weeks, then you have to creep up because the water had been hydrolyzing the calcium hydride you had in your jar. So to be safe for everyone out in the real world, I always say two equivalents. That way they won't come back to me and say, what the hell's the problem? Now, determining the structures of the regioisomer is also problematic. If you look at the NMR, if you look at correlation tables, they will tell you these correlation tables for N and O substitution, they're going to be about the same. So how do you know which one's which? Because the spinach on them are identical. So we had to do to do that, it was actually relatively simple. You do an attached proton test with APT test. In an APT test, a methylene group, a CH2 group, is deflected below the axis of the thing. And so you're able to easily say, this is that carbon there. Okay? Then we did an experiment called HNBC, which is heteronuclear multi-bond correlation spectroscopy, which what that does is give you long-range carbon-hydrogen couplings. It's a two-dimensional NMR experiment, but what it showed you is that this hydrogen here on this methylene group, which we had identified by the APT test, is coupled with this carbon and coupled with this carbon. Same way in this case, that one and that one. But this one also has, this hydrogen is also coupled to the carbon that contains the bromine. This one was lacking, so you knew it was, this one was the inalkylated product, and this is the other. Does that make sense? So now we know how to make it regioselectively. We determined what it was for sure, unequivocally, so everything's good. So this is the new, finished, complete route to reiterate the whole thing. Nucleophilic substitution, electrophilic aromatic bromination, nucleophilic substitution. It kind of rolls off the tongue because it's all things that everyone does when they do organic chemistry. And then an alkylation. Now the nice thing about this, you get an extra level of versatility, and that is you can either install base acid labile acetates such as this one, or base labile acetates such as the methyl. Christian, I'm always blocking your view. See, I can't go this way because I have this cord here. I always have to step over it, and I'm certain I'm going to fall over it sometime. Right? But you can use that as an orthogonal protecting group strategy, either acid or base, sensitive, depending on which group you want. So here's the crux of the matter. We're able to diversify easier in a more convenient way than we had before. And you can, so you can employ various cross-coupling, metal-catalyzed cross-coupling technologies, and they all work, such as Suzuki, Stille, Heck. This is a Heck. This would result from a Heck reaction. Sangashira, that's where you couple alkynes to aromatic halides, or Buchwalds, where you use heteroatoms like these amines. These are all unoptimized. This is just one shot, one try. And so these are all working. Of course, all of these you would definitely not be able to make via the O technology. And if you remember, this one was 30% yield from the O way, and now it's 62% yield this way, and it's done much later. So that's a much, much greater improvement. So then you take one of these guys, and then you finish those last three steps. A D protection, a coupling with this guy, and followed by another D protection. This one takes off the T-butyl group. It's acid sensitive. Couple on this peptide coupling of this amine, and then hydrogenolysis of this protecting group gives you the inhibitor. So you ask yourself, does this work in vitro? And here's the numbers. This one's a 13 nanomolar inhibitor, 9.5 nanomolar, 118. Remember before we were about 200. And we had well, you can see the numbers here, but we have good selectivity too for both enzymes. And I'm going to show you the candidate here in a second. So what's the SAR associated with this? One is that the ring has to be attached directly to the heterocycle. If you add a methylene group, like an extra carbon, like make it benzyl, or 2 carbons in there, you lose potency. If you put something in this right here, at the para position, that's bad because now you can no longer fit in the S2 pocket because you're hitting the back of the enzyme. Okay? 3-aminos is really critical because that's at aspartic acid 60 that we're engaging. You don't want to decrease the basicity of that amine. If you do, you lose potency, and that's what those are. And then we have this other substitution pattern, which you'll see here. That's just summarizing the whole thing. I want to get through this stuff because I want to pontificate on other more interesting stuff here in a second. Here's the crystal structure of it. And so it's 22 nanomolar. It's more than 4,500-fold selective versus 10N, as well as in thrombin. So that's highly selective. But you see we got this extra interaction with this lysine 60A on this insertion loop. That's what's affording this extra selectivity as well as potency. But more importantly, so it works great in vivo. It gives you a pretty picture when you take the crystal structure. But you want to ask yourself, does this work in vivo? That's where the rubber meets the road. And so how do you measure this? Well, you use a bleeding time assessment. No different if you were on Coumadin. You'd go to the doctor. They do these INR values there on people. And this is on a non-human primate. In this case, it was a cynomolgus monkey. It was those little monkeys that the guy spins that crank box, you know, and he goes out with the hat and says, "Give you money." You've ever seen those? Yeah,
Organ grinder.
Organ grinder. Yeah, one of those guys. That's the same kind of little monkeys, and it essentially looks like the old diabetes test where you take that thing and then you measure how long it takes that little prick to clot. Not the new ones; those use much less blood, but the old ones where you get a little bit more. So these 1x, 3x, all these numbers here, those are multiples of an efficacious dose that's been determined what it is via plasma. Okay? So this is our compound, and according to the WHO, you're not allowed to have a bleeding time that's greater than 2x what should be normal. So you want all these things to be below this little dashed line. And all the way up to 4 times the dose that you need, it's limited at 4 because of the solubility of the compound. If you could increase the solubility higher, you could push it. But all 4 of these are all below this little line. That's good. This one, when you go to just a 10A inhibitor, at the actual efficacious dose, 3 of the animals are already above the line. That's a problem. In a thrombin inhibitor, we had 6 of them. Half of them are already above the line at the very first efficacious dose. That's a problem, right? And anything above that, that's showing you that those 2 enzymes have a very narrow window, therapeutic window. And another way of assessing this is looking at how much blood is actually lost at the time. And you actually quantify that by taking the nick that you induced and sopping it up with cotton and then measuring. Okay? And you can see these all stay really, really nice, small, tight together, very little blood loss, and theirs are all sporadic, they're all over the place. It's a little bit more difficult to measure blood loss than it is bleeding time and clotting. Oh, yeah. So what did I do? Developed this polymer assist, this PASP technology, polymer assisted solution phase chemistry, to facilitate development of SAR in a rapid fashion. New cross-coupling technology on these type of templates that hadn't been known before. That technology allowed, incorporation of functionality that was previously inaccessible. This is kind of nice because, if you're in a process plant and you're making, a couple hundred kilograms of this stuff, you don't want to use something like TMS cyanide if there's ever a break or a spill, right? And then, of course, those other 2 in vivo study guides resulted in successful preclinical proof of concept. So I hope I can have just a few more minutes of your time. And I thought what I would do is, this is something I added about 15 minutes before I ran down here. Just show you what's it take to get to the market and the basic paradigm that the FDA requires. FDA requirements for such a thing, okay? So these are all the various stages. This is roughly 18-year on average to do this 18-year process. Up to this point here, this is usually 6 to 8 years. This is usually about a 10-year process. These last two right here. Okay. The first one is called ESD, and that stands for, I don't remember, but something like exploratory screening development. And what this is the base. This is where the idea comes in. Say, hey, my dad takes Coumadin, and when he cuts himself shaving, he continuously bleeds. That's not a safe drug. How do we fix that? That's the idea, and this is where it starts at this place right here. And so it's basic research on disease etiology and pathology. What causes the disease? You try to identify the drug targets. Is there any druggable targets for a therapy, right? You do in vitro assay development and look for biomarkers. If you're going to assay something, you've got to be able to measure it, okay? There's very little wet chemistry involved, but there's actually a lot of chemistry involved. You might have to make substrates, protein substrates, things like this for these guys. The next one's called SDS, and I think that's called screening and design synthesis. Yeah. This is where you employ high throughput screens, not just in vitro screens that you're actually measuring something, but also in silico, fragment-based screening, the stuff that Dr. Darcy does, that's all would be incorporated at this stage right here. Then, from the data that you get out from that, you would do targeted synthesis of a particular compound of interest to see if it actually works and permeates your system properly. And you'd also do early toxicity assessment. If you knock out this enzyme, is it lethal? Right? You need to know that early on because you might not want to be that, might not want to be your target. Choose a different one if that's the case. Then this one's called LD. I think that stands for lead development. This is where you identify multiple chemical series with a minimal in vitro profile, such as potency, selectivity, pharmacokinetics, toxicity. And you define the pharmacophore for your target. And then you begin developing animal and cell models. And then this is actually the first time you look at legal assessment. You want to make sure that you're working on proprietary stuff. You don't be working on a competitor's substrate because if it's patented, then you can't work on it anyway. You wouldn't be able to sell it. This one is called, candidate selection, I think is what they call it. This is where you identify the chemical series, the one that you want to move forward. Hopefully you triage from 4 or 5 chemical series down to 1, right? This is also where you begin to define the pharmacodynamic and pharmacokinetic relationship. Ideally, you want it to be 1-to-1. If I give you X amount, I'm going to see this X amount of result. If I give you 2X, I want to see 2X. Oftentimes it's nonlinear. You can stick in X and you see 4X. And that's always a problem with getting something through the regulatory people. And then you also have to show efficacy in 2 different animal models at this stage. Then this is called candidate selection. This is called, this is preclinical. This is where you've identified 4 or 5 compounds from your chemical series, the ones that have kind of floated up to the top. You do dose range finding, safety in 2 different animal models, and then you also have to define the mechanism of clearance, absorption, distribution. Those all have to be known, or the FDA doesn't allow you to market the drug. You have to know it at this stage. Then you have to do a pharmaceutics assessment. You got to know the form of the material that it is. Amorphic is best. Solubility is better for amorphic material than it is crystalline. You have to have a defined synthetic route. You got to know how to make it. Not only know how to make it, you also have to set up the people who, you have to make sure you have a supply a commercial supply of your intermediates. If they're not out there, you have to find somebody who will do it and abide by that obligation to make sure that it's always there for you. At this stage, roughly only 1 out of 100 projects will make it. Okay? So your failure rate is 99% at this stage. Then you go from preclinical to phase 1 study. A phase 1 study is usually between 20 and 100 healthy volunteers. These aren't people who are sick or diseased in any fashion. They're almost always male. For some reason, socially speaking, women are placed at a higher